So today I'm going to talk about structural bioinformatics and AlphaFold modeling of the human kinome and its interactions. So what we're talking about today are what are called typical protein kinase domains. There are other protein kinases that humans have that have more elaborated folds or different folds. We're not talking about those. So we're talking about the ones that look like pKa, look like EGFR, and other similar kinases. So humans have 493 of, let me use the laser pointer, there we go. 493 of these domains in 480 different genes. So some of the genes have 2 of these kinds of domains. But we can divide these 493 domains into catalytic domains, ones that actually catalyze phosphorylation, and pseudokinases which do not catalyze, they're sort of dead as kinases. So there are 56 of those for a total of 493. The structure looks like this. I'm actually going to switch to a PyMol session. Oops, not that one. So we can look a little bit more closely at the kinase domain. This is actually an AlphaFold model of CDK2 with a substrate-bound. So a typical kinase domain has an N-terminal domain that has some beta sheets. I'm going to try to hide this. Oops. Okay. Has an N-terminal domain that has beta strands and the C, what's called the C helix. The C-terminal domain is mostly alpha helices. In the middle, between those, zoom in, there are 2 loops. One is called the catalytic loop. That's this stuff in green. There's a motif in there, HRD, histidine, arginine, aspartic acid. And the activation loop, which is here in magenta, begins with a sequence DFG, aspartic acid, phenylalanine, glycine, and ends with APE, usually alanine, proline, glutamic acid. So the DFG motif is here. You can see this phenylalanine kind of sticking in underneath the C helix. And here is ATP and 2 magnesium ions. And what's going to happen is the substrate is here in blue, this peptide here. And there is a serine right here that is interacting with the HRD aspartic acid, and that is what is going to get phosphorylated. It's going to get phosphorylated. This terminal phosphate group is going to get covalently attached to this serine right here. And so these are the different parts of it, and what we're going to look at today are what makes this kinase active as an enzyme, or when is it inactive, and how does this domain interact with other domains and interact with other proteins. And so we're going to focus on the conformation of the activation loop, because that's really what makes a kinase active or inactive, and because that's what interacts with the ATP and the magnesium, as well as the substrate. So go back to this, make sure it shows up, okay. So I've already talked about all of these different pieces. I won't do it again on here. So modeling the human kinome, the way we approach this problem and many other problems in cancer biology or in structural biology is first to look at structures in the PDB, experimental structures that are X-ray structures or cryo-EM or NMR structures. Then we use information that we get from those structures to think about, well, how would we model these structures for all of the human kinases, not just the ones that are in the PDB, using AlphaFold. We may use other experimental data that are available, mass spec, mutagenesis, NMR, to provide more information. And then it's possible to take the AlphaFold models or the structures in the PDB and do molecular dynamic simulations where we watch the protein change structure. We don't do a lot of that in my lab, but the structures, the models that we build, other people are using them for simulations. So the topics I'm going to cover and the things that we are working on is, first of all, to think about what makes the kinase active or inactive. And we want to know that because in a lot of cancers there are mutations in the kinase domain that make the kinase constitutively active. It's always active. And many of the drugs that people take for cancer bind to inactive forms of the kinase, or they may bind to active forms of the kinase. And we need to know how the mutations that they may get while they're taking a drug that make the cancer resist the drug, what are those mutations doing? Are they forcing it to be more active? Are they preventing it from binding the inhibitor in the active side, or is it some kind of allosteric mutation? We want to look at the structures of the full-length proteins. So, oops, oh, don't do that. There we go. I learned very recently that you can, on a Windows machine, you can do Control L and the little laser pointer appears. On a Mac, it's the Command L. I was just at a conference and they said, oh, if you do this little shortcut, you get the little laser pointer, which you can then move around your screen. And I thought was the best thing I learned at that conference. So we want to look at the domain structures of the full-length proteins. Many kinases are 500, 1,000, 2,000 residues long. And not only do they have the kinase domain, but they have lots of regions of disorder, and they may have other domains that interact with the kinase domain, or that interact with substrates, or that help localize the kinase in different places in the cell. So we want to know where those domains are. Do they interact with the kinase domain? Do they interact with each other? We also want to model how kinases interact with other proteins. Their substrates in particular, but also other proteins that regulate the kinase, either where it is in the cell or whether it's active or not, how it interacts with other proteins. And then finally, we're using protein design. We and other groups are using protein design to design small proteins that might inhibit a kinase or relocate it. So in 2020 or in 2021, our world changed, the world of protein structure prediction, the world in structural biology in general, because of the appearance of a program called AlphaFold, which I'm sure many of you are familiar with. What happened in December of 2020 was the CASP experiment. Every 2 years, there is a competition for predicting structures of proteins. So the organizers gather sequences of proteins whose structures are not yet publicly available. They're being determined. And they ask the prediction community, the computational biology community, submit models of these proteins by certain target dates. And then when the experimental structures come out, we will assess them. We will assess how accurate the structure predictions are compared to the experimental structures that are then available. My group were these assessors. So those people are called the assessors. We were the assessors at CASP 6 and CASP 11. So 2004 and 2012, I think, is the other one. And so we were one of the groups that actually assessed, told people, this is the top ranked group, this is the next group, in terms of how well people did predicting structures. The y-axis, so the x-axis here is the year. It started in 1992, and it's every 2 years, so there'll be one later this year. And the y-axis is how accurate in percentage terms the predicted structures are of proteins that were not at all in the PDB. Nothing related to these targets were in the PDB. These are sometimes called the de novo targets or ab initio structure targets. There were no templates, no related structures that were available at the time. That's the really hard thing. Sometimes people do comparative modeling when you take one kinase and model it on the basis of another kinase, but those folds are very similar. These cases were all brand new. They were not in the PDB. And you could see that in those very early years, it was 10% down here. For a decade, it was, sort of hovering around 30%. You really couldn't do very much. Started to go up, and the blue curve is without AlphaFold, without the contribution from the company called DeepMind. And it was heading, towards maybe 60%. So some of the structures were right, some of the structures were wrong, maybe some of the structures were partial. But AlphaFold came along, and in CASP14 in 2020, that group were getting about 90% of the structures correct. That's almost as accurate as experimental structures, because if you compare two different structures of the same protein, there may be conformational changes, so that maybe only 90% of it is the same. So that's almost as accurate as experimental structures. So it was really exciting and kind of changed the game. These are two structures, one of which was an ab initio, a de novo structure prediction, and the other one was based on a template. Both of these came from the AlphaFold group. It's the one on the left, this more complicated fold, that there was no such structure related to it in the PDB at the time. This is in 2020. And you could tell this is a very complicated looking protein. So it wasn't just a little 3-helix bundle, it was a complicated fold that they were able to get the correct structure, de novo. The one on the right has a template, it's easier. So how do these deep learning models work? I just want to give a very brief kind of description so that you understand how deep learning works in any kind of AI that you might come across. So particularly for AlphaFold, it learned from all of the structures in the PDB, plus it did multiple sequence alignments. They found lots of sequences that were related to each protein in the PDB. So if it's a kinase protein, lots of other sequences of kinases, and made a multiple sequence alignment. AlphaFold is fundamentally a knowledge-based inference system. It doesn't use any physics equations. It doesn't use any of the principles of physics that we know. So it takes a query sequence, it takes the multiple sequence alignment. It can take experimental structures that are related to the target and use the distances within those as extra information to go into the network, and then it predicts the structure. So any neural network works something like this. You have inputs, which probably are a series of numbers. It could be the pixels in a photograph, for instance, what color each pixel is. It takes that input and gives it to a mathematical function, and that function is a function of those inputs, but also some numbers that are called weights. We'll call them W. And those are just numbers that will, and somehow be multiplied, and lots of mathematical functions will operate on those input coordinates, and then the output is something Y. Y might be the predicted structure of the protein. If it's a photograph, Y might be this is a mouse, a dog, or a cat, something like that. So if it's a mouse, a dog, or a cat, that's called a classifier. If it's a number or a big set of numbers, that's often called that's a regression, effectively. And the network has to be trained on real data. It has to be trained on real x1, x2, x3 sort of input things that are easily measured, and then output Y may be something that is not so easily measured. Like doing an experimental structure takes a long time. Getting a sequence of a protein is very easy. So for AlphaFold, the input is the sequence, the multiple sequence alignment, and the output is the structure. And so we need to train these numbers W. And there are three tricks that are used to train the neural network to do this procedure. The first one is simply to take inputs like x1, x2, x3, and multiply them by numbers and then add them up. So z1, I'm going to get z1 and z2 out of x1, x2, x3. I'm going to multiply them by three numbers, w11x1 plus w12x2 plus w13x3 gives me a new number, z1. And I do the same thing for z2. In mathematical terms, that is multiplying a matrix times a vector, which gives you another vector. And that vector can be smaller or bigger or the same size as the input vector. The number of numbers in z1, z2, z3, z4 can be different than the number of inputs. You just have more bigger matrix. The second trick is to apply something called an activation function. And what an activation function do is it takes those numbers z1, z2, and those numbers can get very big and negative, very big and positive. And the network is more stable if the range of those numbers is relatively small. So what the activation functions do is they turn them into a number between 0 and 1. There are different functions, we call them sigma, or between -1 and +1. So even if the number goes off to infinity, plus infinity, the number will then, after the activation function, will come out nearly 1.0. Goes off to minus infinity, it'll be minus 1.0 and everything in between. And so these are the different kinds of functions that are used. The third trick is to stack multiple layers of these, doing the dot products with the weights and then applying the activation function. So here we have x1, x2, x3. We're going to apply some weights WA. We're going to apply a particular sigma. We're going to take those numbers and input them to the next layer. We have another set of numbers, WB, and we have another set of sigmas out to WC, out to WD. So this is 4 layers. And as I said, the size of the vector can go up and down. And so, here it is maybe slightly bigger than the inputs. It's much bigger, same size. Now it's gotten smaller. And then the last thing is multiply it by a set of weights and you get the output. And the way the training is done is basically, given some x1, x2, x3 experimental and the output y experimental, change these weights so that the output y looks, from the network, looks like the experimental y, let's say the protein crystal structure. And it uses derivatives and calculus to say, oh, all right, if I get a y that is quite different than the output, than the experimental y, I should change the w's in particular. These w's should go up, these w's should go down, and then I'll get a better y. And you just keep doing that over thousands of iterations, and you get w's that do a good job of giving you the output y's compared to the experimental y's. There's another trick which are called diffusion neural networks, a particular kind of network. This is relevant today because AlphaFold 3 uses a diffusion neural network. And what a diffusion neural network does is it is a denoising kind of thing. It's like taking a blurry photograph of your great-grandparents and making it a nice, sharp, good photograph of your great-grandparents. And the way we do this for, let's say, for protein structure prediction is to take a lot of experimental structures, real crisp-looking structures, and add noise to them. Just add random numbers to every coordinate, step by step by step. So the first one looks similar, but the next one more different. And by the time you've done it quite a few times, you've got just a bunch of atoms everywhere in space, and it's just a blob. And now you can use that kind of information, that set of structures from nice and crisp to really messy, and train the network to do the process in the other direction. To take, a messy random set of atom spheres and slowly denoise them step by step until you get an experimental structure at the end. And you add constraints. You tell, this noisy set, you say, well, it's this noisy set, but it's a protein of length 100. It has this particular amino acid sequence. You might have a multiple sequence alignment. You can add information. So with that extra information, the network knows, okay, I'm going to denoise it, but I'm going to denoise it knowing what it is, knowing it's 100 residues long, knowing what the sequence is. And that's what you train the network to do. So AlphaFold 2, the original one that came out in July of 2021, was just for single proteins. It was trained on single chains of proteins, and it predicted the structures of single chains of proteins. But later in 2021, the same team at the company DeepMind came up with a program called AlphaFold Multimer. And AlphaFold Multimer was trained on complexes of proteins, 2 proteins, 3 proteins, 4 proteins, in the PDB whose structures have been determined. They could be homodimers, heterodimers, larger complexes. But it was trained to predict the structure not only of each protein, but how the 2 proteins interact. So the first version was November 2021. They had a couple more versions. The last one is 2.3 from December 2022. That's the only one we use. We don't use the earlier versions. It was trained on a much larger part of the PDB, and it had some other fixes that helped it give the right answer more often. Just as an example, this is a structure that we predicted the structure in February of 2023. The complex of the purple, the blue, and the green protein was already known. That was an experimental structure. But my colleague Xavier Gagne at Temple University was very interested in how does this inhibitor protein, this is a phosphatase, an enzyme that takes phosphates off of proteins. That's the purple protein. The blue protein helps pick substrates for dephosphorylation, and the green one is sort of a scaffolding protein that holds them all together. But we had no idea what the structure of this piece was. And so we did the calculation, and we got this 2 helical structure. The active site is right here. This is these are manganese ions in the active site. And my colleague did lots of experiments to prove that this model was very likely correct. 6 months later, in October of 2023, the experimental structure of this complex came out, and it is virtually identical. So in other words, AlphaFold 2 was able to put this protein in exactly the right spot. There's very little difference between that experimental structure and that predicted structure. AlphaFold 3 came out in 2024. There's a AlphaFold 3 server anybody can use. It's free. It's paid for by Google. Google has billions and billions of dollars. So they have lots and lots of computers, and you can use their computers for free. You just put in sequences. You can add lots of other information. So this is, again, that diffusion idea of taking a blob of atoms and then denoising them into this trimeric structure, each of which has 2 lobes, so you get this sort of hexamer looking thing. AlphaFold 3, unlike AlphaFold 2, can do models of proteins with DNA, with ions, with ligands like ATP. And so on the left is a predicted structure of p53, 4 copies of p53 bound to DNA. So this is a transcription factor, p53. On the right-hand side is a small ligand bound to p53. The crystal structure is in pink, so that's this pink small organic molecule here. The predicted structure with a program called Boltz-2, which is very closely related to AlphaFold 3. AlphaFold 3 you cannot use for any commercial purpose. Even in the future, like if we did a structure prediction now and we wanted to eventually commercialize some molecule, we might get in trouble if we used AlphaFold 3 along the way, and nobody wants to get sued by Google because their lawyers will destroy your lawyers. And so this predicted structure is in magenta, and you can see that it's very similar, that it was able to put the molecule in the right place. I will say that if you try to predict the structure of a protein with a small molecule ligand, if that ligand is not like anything in the PDB, if that protein structure is not in the PDB, or maybe it is, but there is no ligand bound at that site in any of the structures of that protein family, AlphaFold 3 and Boltz-2 can get the right answer maybe 20% of the time. So it's not very good. I mean, to do stocking small molecule ligands is very hard. And there's not enough experimental structures to train the network to do a really good job of that all of the time. However, these programs are better than some of the old-fashioned docking programs at docking ligands. If it is related to something in the PDBs, a kinase, for instance, you might get the answer right 80 or 90% of the time. That's an easier problem, but it does do a good job of it. So the way the programs work is like this. It takes a query sequence. You get a multiple sequence alignment. You can search the PDB and get templates. You run AlphaFold. It's got those weights. It's got those sigmas. And then it outputs a set of coordinates. And it outputs a bunch of scores that are going to tell you how good it thinks the model is, how likely is it that that model is correct in various details. So I want to talk a little bit about how those work. The first one is something called PLDDT, which stands for Predicted Local Distance Difference Test. It's a bit of a mouthful, but it works like this. During training, they have experimental structures like this. This one on the left is a kinase, Aurora kinase. The one on the right is a predicted model at a certain stage of the training. And the PLDDT is related to, well, if I take, PLDDT is assigned to every residue. Every residue has one number, PLDDT. So let's take this one. And what I do is I measure the distances within a 15 angstrom radius around that atom. I'm only showing you a few of them, of all the other residues that are nearby within, say, 15 angstroms. And so I've given you 7 of those distances, 5.5 angstroms, 12.1 angstroms, et cetera. That's the crystal structure. Now I take that same list of distances and I look inside the AF2 model and I measure the same distances at some stage of the training. And I compare this set of distances with this set of distances. And so the differences between those are, if I start up here, this one is 5.7, this one is 4.8, that's a difference of 0.9. The next one is 6.2, and the one over here is 7.2, that's 1.0. And so I measure all of the differences between the distances of the crystal structure and the predicted structure. The LDDT, if I was actually going to say what's the experimental LDDT, it's the number of residues that have distance differences better than 0.5 angstroms, 1.0 angstroms, 2.0, and 4.0. So I measure what percentage of them are better than 0.5, what percentage of them are better than 1.0, better than 2, better than 4, and it's the average of those 4 percentages. Seems like a strange number, but it works reasonably well. And so the answer is, well, 0 out of 7 are better than 0.9, 0 out of 7 are not, 0 out of 7 are better than 0.5, 1 out of 7 is better than 1.0, 5 out of 7 and 7 out of 7 are better than 2 and 4 angstroms. That comes out to 46%. So that's the number that would get attached to that amino acid. And the network has been trained to predict this percentage, to predict 46%. Because it has been trained on real experimental structures, and it knows how well it is predicting the structures. You can compare them, and it's been trained to predict that number. So you get a model of an AlphaFold structure, and every residue has a PLDDT attached to it. What I've done below is this is a kinase that has 2 different domains. Orange is the kinase domain. On the left-hand side, I've colored all the residues where the PLDDT is worse than 50. So really only 50% of the atoms are really in the right place in the neighborhood of that amino acid. And these are all the disordered regions, these big loopy things. On the right-hand side, I've colored it by the PLDDT. So the dark blue has really high PLDDT, 80, 90, near 100. The red regions have very low PLDDT. And you can see the folded domains have good PLDDT, and the loops in between, some parts of the structure are green, but all of the disordered regions are red. So anytime you get an AlphaFold model, you want to color it by the PLDDT. Inside PyMol or Chimera, it's in the B factor field, so you say color gray, comma, B less than 50, and all of the disordered regions will turn gray. So we want to be able to model protein-protein interactions. Remember I talked about AlphaFold multimer. And so this is a complex of 2 different proteins. One of them is RAF1, which is a kinase, but it also has a RAS binding domain and a cysteine-rich domain. So that's in pink and magenta. That's one protein, plus the disorder that, so here's kind of the domain diagram up here. And then the other protein is KSR1. It's a pseudokinase, inactive kinase, but also a kinase domain. And it has a coil SAM domain, also a cysteine-rich domain. And so it's the blue kinase domain here, and the green and this sort of yellow, I guess, right there. There is a score called ipTM, which comes from AlphaFold, output by AlphaFold. And it's a measure of how accurate it thinks the protein-protein structure model is, the interface between the two proteins, the relative orientation between the two proteins. And the score I get for these two full-length proteins, one is 600 residues long, the other one was 900, is 0.38. However, if I isolate the sequences of just the two kinase domains, the magenta domain and the blue domain, ipTM jumps up to 0.82. And in fact, this structure looks very much like a homodimer of RAF1 or BRAF. It's not in the PDB, this structure, but it is almost certainly correct given what we know about how KSR1 interacts with RAF1. So this structure, let's say the structure is correct. It's not helpful though that if I do the full length proteins, I get 0.38, kind of low score, it's below 1/2. And if I isolate just the interacting domains, the score goes way up to 0.82, 82%. Why is this and how could we fix that? That's not helpful when we want to use this program. So this score is based on something also that AlphaFold puts out, and it's called the PAE, the predicted aligned error. So let's look on the left-hand side. What does that mean? If I align a predicted structure and an experimental structure on one residue, just the N, C alpha, and C atoms of one residue, so that's what I've done here. It's the two magenta side chains. This is aspartic acid and a kinase. I've aligned the N, C alpha, and C. Now I'm going to look at another residue in the structure. And in the PDB, that position is here. In the model that AlphaFold is predicting, it's here. And so there's a distance between those. If I'm aligning on this residue and I'm looking at the PDB structure, the residue is here, and in the model it's here. What's the distance between those? That's called the aligned error. And so the network has been trained to do that. So for every residue that it aligns on, we'll put that along the y-axis. I'm going to plot in different colors the predicted aligned error of all the other residues. So again, this is two chains. So this is RAF1 versus itself. Here's the kinase domain, this blue square. Here are the other two little domains, these little blue squares up here. This lower square on the right-hand side is KSR1. Again, the kinase domain versus itself is blue. Those are low PAEs, 2, 3, 4, 5 angstroms. And the other two domains here. And all the red is all of that disordered stuff, versus each other. But also, the kinase domain and the CRD domain, there's nothing blue here in this region. So in fact, the position of the CRD relative to the kinase is very uncertain. These off-diagonal rectangles are the relationship of one protein to the other protein. And so you can see here, this square here is the kinase domain of RAF1 versus the kinase domain of KSR1. And it is very confident about that interaction. Remember, this is a kinase-kinase domain interaction. That's the part that I think is correct from what I know. Whereas there is very little interaction of, say, the CC-SAM domain with this other domain up here, or the CC-SAM domain with the kinase domain. So there's very little interaction of the other domains or the disorder between the proteins other than kinase domain to kinase domain. And it's not quite symmetric, but it's almost symmetric. Here's kinase-kinase domain, RAF1, KSR1. Here's kinase-kinase domain, KSR1, RAF1. So why does this ipTM not work very well? The ipTM is, okay, if we look at one residue at a time in let's say RAF1, and I look at all of the residues at KSR1 and how good are the PAEs? And I'm going to transform the PAEs to a number between 0 and 1. So 0 means not at all certain, really big PAE. PAE could be 30 angstroms. 1.0 would be, it's really certain where the aligned error is. I'm going to make it, a number near 1. And so what the ipTM does is says, okay, over all the residues of KSR1, I'm going to evaluate how good are the PAEs. Same thing here. If I go downwards and look at KSR1, I'm going to look at all of the amino acids in RAF1. And I'm going to calculate a number between 0 and 1. The problem is, if I look at all of these residues here, there's a lot of red. So I'm taking an average number over, half of the residues are 30 angstrom PAEs. They're really bad. So I'm averaging, 300 of the 600 numbers are something like 0.1, a very low number for this ipTM. Half of it is the kinase domain, kinase-kinase interaction. Those are all 0.8, 0.9. And so I'm going to get a number that is averaging out to about 0.4. Remember I said this interaction is 0.38. The way we fix that is instead of calculating this over the entirety of the other chain, I'm only going to evaluate it over the part of the other chain that AlphaFold is certain about. So if I start with RAF1 somewhere in the kinase domain, and I look along the x-axis, all the residues in KSR1, but I only evaluate this function over the part that it is certain part, the part that's in blue. And so now I get a score, like 0.7 or 0.8, because now I'm only evaluating this function over the part that AlphaFold is certain about. And I use a cutoff, a fairly generous cutoff of 10 angstroms that says, as long as the aligned error is within 10 angstroms, I'm going to evaluate the function there. The same thing down here, if I align on KSR1, I evaluate all of RAF1, I get a good score. So for this complex, remember ipTM was 0.38. Of just the two proteins, it was 0.82. But this ipSAE score, ipSAE is what we call it, is 0.75 for both. So that's good. It's behaving the way we want it to. We did a sort of benchmark where we took 40 true complexes from the PDB and 70 false complexes from the PDB that were basically scrambling the true complexes that were all human proteins. And the score here is on the x-axis. This is like a histogram, just the density, the frequency. And so the blue curve is the true complexes, and the false complexes are in the magenta curve. And ipTM, there's a lot of overlap in the middle. There's a lot of both true and false complexes that are getting scores between 0.4 and 0.6. That's not helpful. On the right is our score, this ipSAE score, and there's very little overlap between the blue curve and the magenta curve. So either the false complexes are almost all less than 0.1, the true complexes are almost all greater than 0.4. There's a little bit of here, there's a few true complexes that AlphaFold doesn't score well no matter what. So this is a better score for distinguishing true interactions from non-interactions. It comes from a Latin phrase, res ipsae loquuntur, which means the things themselves speak. The singular of that is res ipsa loquitur. I took Latin in high school, so it's fun. And in legal terms, in English common law and in American law, there's a similar law, a similar concept in French law. It's not called this. But the res ipsa loquitur idea is if you see a car accident, then somebody must be at fault. Something must be negligent. Something went wrong. So the thing itself speaks. And in this case, I wanted the AE for aligned errors. That was the trick. It is being used in the AlphaFold database, which is online. You can get predicted structures of 200 million proteins in UniProt. They are now doing homodimers and heterodimers of proteins in certain genomes, and they are using the ipSAE score above a certain value to say, this is a likely homodimer, so we're going to give you the coordinates of the model. If the ipSAE score was less than 0.6 is the cutoff, they're not going to give you that homodimer model because they're very uncertain about it. I'll skip that. It is also being used in protein design. I'll get to this a little bit later. But the idea of protein design is you have a target of a protein, target protein that you want a small mini protein, peptide or 60 residue protein to bind to it, and you use a diffusion neural network to design a protein that will bind to it. And I'm not going to go through these papers, but these 3 different papers used our ipSAE score as a good score for determining true binders from non-binders. You do 10,000 designs on the computer, you're only going to make 10 or 50, how do you rank them? And our ipSAE score is turning out to be very useful for this purpose. So back to kinases and the active kinases. I've already shown you what the active site looks like of CDK2 and the substrate and how it interacts with the ATP. Okay. We can think of kinases having a landscape of different conformations, one of which is active and the rest of which are inactive. So under APO conditions, no ATP, maybe there's a conformation of the protein that is inactive that we're going to call I-APO. If I add ATP, but the kinase is not yet active, I might get an inactive form of the kinase bound to ATP, and now that's the most stable version. If I phosphorylate the activation loop, that's how many kinases are activated, or other proteins come and interact with the kinase, and that activates it, now the active structure is the lowest in free energy, and that's the structure that you're going to see. You might also add some weird inhibitor, class of inhibitors, and they stabilize yet a fourth conformation that we're going to call I-weird for weird inhibitor. So for example, for a kinase, the APO structure looks like this. Here's the activation loop in orange. When ATP is bound, but the kinase is not yet active, that's what's here in blue. It's phosphorylated, but it's still not active because it needs a protein to bind to become active. When this protein is bind, it's called TPX2, this little green helix here. Now the kinase is active. You can see the activation loop goes out, it comes back, the substrate would fit here. It looks very much like this structure, where the loop goes out and comes back. That was CDK2. And then here is a set of weird inhibitors that stabilize. Now the loop goes way to the left and then comes back. So that's yet a different structure. And then these are a whole bunch of other structures of the same kinase. So there are two factors that make a structure active, or two things that it has to be able to do. The first one is it has to be able to bind ATP and magnesium ions. The magnesium ions are required for catalysis. And so that's the site around here. Here's the ATP, here are the two magnesium ions. And they interact with kind of the first part of the activation loop here in pink. The second thing the kinase domain has to do is it has to bind the substrate. And here's the substrate kind of in a rainbow color from left to right. And it sits kind of, in between parts of the activation loop. Here's the N-terminal part of the activation loop. Here's the C-terminal part of the activation loop. And the substrate kind of sits between. So we have to look at structures and find out what would be required for these two different things. And so what we did is we took from the PDB 54 different substrate-bound structures, structures that had a substrate bound in the active site. Most but not all of them also had ATP and magnesium. And they look something like this. So these are the different families of kinases, AGC, CAMK, CMGC, the different subsets of kinases. And in magenta is the substrate. The activation loop is here from blue all the way to red. And in most of the families, there's this structure here towards the C-terminus that looks like take a round bottom boat and turn it over, as if you capsized while you're riding a boat on a lake. And that structure is common to all of these substrate-bound structures. Tyrosine kinases are a little different. They bind the substrate in a different way. Many of the structures that we included here are autophosphorylation complexes, and those are structures in the PDB where one kinase is sitting in the PDB, but there's a neighbor inside the crystal of the same kinase where some site that usually gets phosphorylated is sitting in the active site of this one. So this kinase is acting as the substrate, this one is acting as the enzyme, the kinase, and it's going to phosphorylate that one. And those are kind of snapshots of phosphorylation sort of in progress. And we did a paper on this more than 10 years ago where we found a lot of these in the PDB. Some of the authors had recognized that this thing was taking place inside their crystal. Some of the authors had not. But we did it in a kind of bioinformatics way where we looked at all 8,000 structures of kinases and looked for all of these interactions. So I think 17 of these 54, about a third of them, are from these autophosphorylation examples. This is by far the biggest set of substrate-bound structures that anybody's ever written down in one place. I've seen papers that use 10 of these, but we get 54 of them, partly because of the autophosphorylation. So the first thing I said has to bind ATP and magnesium. That's what this site looks like. Here is the substrate that's going to get phosphorylated. Here is that aspartic acid of the HRD motif. There's a hydrogen bond here. And this is what ATP looks like and the beginning of the activation loop. So remember it's the DFG motif. This is X residue is the residue before the DFG motif. And you can see that the ATP and the XDFG all look the same across this set of structures that are about to phosphorylate. And so what we do is we look at, first of all, the position of the phenylalanine and the aspartic acid, but then also the dihedral angles. If you're familiar with protein structure, you know that the backbone has 3 different dihedral angles called omega, phi, and psi. Omega is almost always 180, so we usually ignore it. But it has phi and psi, 2 different dihedral angles that tell you which direction the backbone of the protein is going to go in as you follow the chain. And for these XDFG motifs, what happens is that for all of them, the X residue is this is called a Ramachandran map, which you may be familiar with, the phi dihedral on the X-axis and the psi dihedral on the Y-axis. These are numbers between minus 180 and plus 180. For the X residue in blue, they're always up here, up in the upper left. For the next residue, which is the DFG, the ASP, they're always over here on the right-hand side. The phenylalanine is all up here, sort of in the middle on the left-hand side in orange, and the glycine is here just below it in red. So they all look like this, and these regions of the Ramachandran map have names. This is the A region because alpha helices are usually contained here. This is the B region because beta sheet residues are contained here. This is the L reason because they're for left-handed alpha helices. And E is for epsilon or extended, you could think of it. Also, the dihedral angles of the side chain of the phenylalanine here and of the aspartic acid here. The side chain has to go in particular directions in order to bind ATP and magnesium properly. And so the aspartic acid has the first dihedral angle is around 180. The phenylalanine is around -60. So this is called a trans rotamer. This is called a G- rotamer, -60. So these are the requirements for binding ATP and magnesium. There's one more requirement, actually two more. There is a salt bridge in the N-terminal domain. That's this glutamic acid here, this lysine residue here. It's very well conserved in all kinases, just about. And that helps to bind the magnesium, and it helps to bind the ATP. And so that salt bridge has to be formed, that hydrogen bond between a positively charged residue and a negatively charged residue. So that's how the ATP is bound in magnesium. What about the substrates? So we looked at these 54 structures. We figured out, okay, what are the contacts between amino acids in the substrate and amino acids in the activation loop in the kinase? And so on the x-axis are the residues in the substrate. P in the middle, that's the site that's going to get phosphorylated. P plus 1, 2, 3, 4, 5, those are the residues that are C-terminal to the phosphate site. And P minus 1, minus 2, up to minus 5 are the ones before it, N-terminal. And the activation loop is here. So the activation loop starts with that DFG motif. DFG1 is aspartic acid, DFG2 is the phenylalanine, the glycine. The fourth residue of the activation loop, DFGX, whatever X is, has lots of contacts with the substrate residues P, P plus 1, P plus 2. These are the non-tyrosine kinases on the left and the tyrosine kinases on the right. So again, they bound a little bit differently, but they have that DFG4 residue in common. Down here is counting back from the end of the activation loop. Remember that was the APE sequence. So here's the E, here's the P, here's the A, and now you're going backwards in the sequence. And around APE 5, 6, 7, 8, 9, all the way up through about APE 12, there are lots of contacts with the substrate. And the contacts are I won't go into the details, but the contacts are somewhat different, similar, but somewhat different between non-tyrosine kinases and on one hand, tyrosine kinases on the other. So we came up with a bunch of dihedral angle criteria and distance criteria that help us separate, okay, these are substrate-bound structures from non-substrate-bound structures, from other structures that we see of the same kinases. And I'll just give one example. This is aurora kinase with that TPX2 protein. This is the active form of this kinase. This TPX2 protein in orange is what activates aurora, so that's how we know that it's active. Here's ATP. I can measure a distance. It's a little bit hard to see, but it's between the 9th residue back in the activation loop, so that's this cyan color here, this light blue, to a residue next to the HRD motif, or part of the HRD motif, this arginine. And that's a very short distance in active structures. In all of these substrate-bound structures, it's sort of 3-4 angstroms. In this inactive form, these are the structures of aurora that are inactive. The activation loop is kind of this double hump on the back of a camel. And this distance is more like 10 angstroms. It's very far apart. If I superposed a structure with a substrate here, this loop would bump into the substrate. The substrate would not be able to bind. The active site up here has ATP in it. This motif, the ATP magnesium binding site, is perfect. It looks exactly like active structures look. But the C-terminal part of the activation loop is very different, and it would not bind substrates. So we came up with a bunch of criteria for ATP and magnesium, and then for substrate binding. They're based on dihedral angles, and they're based on a bunch of distances. I won't go through all of them. And so we come up with these criteria, we can apply that to today's PDB. Remember I said there were 437 different catalytic kinase domains in the human proteome. In the PDB today, only 111 of those 437, about 25%, are actually in the PDB in their active form. If we apply these criteria to the PDB, we only get a quarter of the kinases do we actually see in the PDB as active. If we take our criteria and say, okay, apply it to the whole PDB, we look at all the active structures. That's the graph on the left. This is one of the dihedral angle pairs that we used, residue 8 from the end of the activation loop. Well, in the active structures, the points are all here. In inactive structures, well, some inactive structures have, are active by that criterion, but all the red points are, would be inactive because of this one criterion on residue 8. The first row are the non-tyrosine kinases, the second row are the tyrosine kinases. The same thing for the distances. This is one of the distances that we use, but the one between residue 9 and that arginine. In the active one, they're all better than 4 angstroms in the non-tyrosine kinases. In the inactive structures, yeah, some of them are active by that criterion, but many of them are inactive by that criterion. So this is just we did this for all of our different distances and dihedral angles. There's a preprint online, but also the paper will be coming out soon in Biochemical Journal. And we have graphs for all of the different dihedral angles. So then we want to say, well, can we model all 437 kinases with AlphaFold? Because it would be really nice to have active structures of, well, we have 111 out of 437. That's 326 kinases, I think if I did my math right, that we don't have active structures for, and we'd really like to know what those look like. So we take the query sequence, we use ortholog sequences of each kinase that are more than 50% identical to each target and not similar to each other, 90% sequence identity or less to each other. We search our kinase database, and we have a whole bunch of templates, PDB structures that we know are active by our criteria, and we use those as information to put into the network so that AlphaFold is convinced to make active-looking structures. And we give it the multiple sequence alignment, and then we run AlphaFold many times, and then we get an active structure. We get a whole bunch of models. We run it many times. Some of the models will be active, some of them won't be. But we use our criteria on the models and say, okay, these are active models. So now we've made active models of each of the kinases. And we use our ipSAE score, kind of an intramolecular version of it, where we score the activation loop by aligning on residues in the rest of the kinase domain. Remember, you align on one residue and you score the predicted aligned error to all the other residues. So those residues are in the activation loop. That's our ipSAE score intramolecular version of it. This is sort of what it looks like if on the x-axis, for these are a whole bunch of kinase structures that we have substrate-bound structures for. The x-axis is the ipSAE score between 0 and 1. The y-axis is the RMSD of the activation loop of the experimental structure to our predicted structure. And you can see this sort of almost always a very poorly scoring models, have bad RMSDs. High scoring models on our ipSAE score have good RMSDs. So it says that our ipSAE score is not only, we apply this only to the active models, but some of the active models are not so good. They don't score very well. The best scoring active models in the activation loop also have good, they're closely, they look a lot like the experimental structures of the same kinases. Okay. So this is sort of what they look like for each of the different families. Remember I said that upside down bottom boat that we see in all of the structures, except the tyrosine kinases are a little different. And all the models are available online. I'm going to skip this section in the interest of time. All of these models are on our KinCore database, which stands for Kinase Conformational Resource at this address. And there's a table for every kinase and for every family, and it has the PDB codes. It has our AlphaFold models. That's what these first, I know you can't read this, it's too small. And all of those measurements that we use to determine active and inactive. Also tells you what the ligand is, what kind of ligand it is, mutations in the kinase sequence, whether it's phosphorylated on particular residues. So if you're interested in any one kinase, you can go here and find all of the available experimental structures for that kinase and examine them. The next project I want to tell you about, I might shorten this a bit, is remember I said we were interested in the different domains of very long kinase sequences. It might be 2,000 residues long. If every time you read a paper about a particular gene, the first, and the protein made from it, the first figure always has a little domain diagram that says, oh, there's a kinase domain here, and a pH domain here, and an SH2 domain here. They are messy, they are inaccurate, they are ugly, frequently. And here are just some examples. Kind of bad colors and a little hard to read, little funny symbols. And sometimes they're missing pieces of information that we know to be true. So for this one, for instance, there is a ubiquitin domain in this region, and the authors of this paper completely missed the ubiquitin domain. They also don't do them to scale, and they often don't put the residue numbers, so you can't tell where does this domain begin and end in the sequence. They just, it's just a kind of schematic diagram and not a very good one. So we made models of all of the 480 proteins containing kinase domains, whether they're pseudokinases or catalytic ones. So here's just examples. And I colored the domains, in each one in different color. With the orange is one of my favorite colors, so I did the kinase domains in orange, and all the other domains. And so we took these structures, we used a database called ECOD, which stands for Evolutionary Classification of Domains. It runs kind of automated code and identifies domains in the protein structures in the AlphaFold database. So we started with those assignments and colored by those assignments and said, okay, there's a kinase domain here, there's a pH domain here, there's an immunoglobulin domain here. I spent months, a few hours every night, a lot of hours on weekends, much to my partner's chagrin, and wrote down where the domains were according to ECOD, and then I would look at the structure and go, yeah, I would make that domain a little bit longer, or there's a piece of structure over here that's a nice little folded domain, and ECOD doesn't have anything. So that might be a novel domain that we want to identify and give a name to. I thought this was a blast. I thought this was lots of fun. Now, I'm not sure a lot of people would, but I spent 20 hours a week for 15 weeks looking at kinase structures, and I thought, well, this is really fun. So, but these extra domains might be activating, they might be inhibiting, they might bind substrates, they might locate the kinase in different parts of the cell. So I'm going to go to here, where then we were able to write some code that made these domain diagrams for all of these kinases that had all of the information from my list of assignments. And we tried to choose nice colors. I don't think we always succeeded, but some of the colors are nice. And in 480 proteins, I identified 1,637 domains. Again, I'm probably the only person who's crazy enough to write down 637 sets of numbers. We also looked at every kinase domain. Often there is a tail that binds right nearby sequence that binds to the kinase domain. I wrote those down as well and called it NT tail. We came up with consistent names across closely related kinases. The names in ECOD and some other assignments sometimes are all over the place. Sometimes people use synonyms, very similar names, or similar names or different names for the same thing. We always have the same thing is always called a particular thing that is observed in multiple kinases. They all look like each other. We give one name to. So we're very particular about how to name things. And that's often, that's the way humans work, right? We use words to describe things. It's good if we use the same word to describe the same thing every time we see that thing. That's not quite the way language works, but it's easier to understand if one word is used for one kind of thing, and that's what we did here. So for example, these sets of kinases up here have these long coiled coil regions, these big long helices in blue and in green. And they're very similar in sequence and in structure across the different members of this family. In ECOD and other sources, these things are called all sorts of different things, some of them not correct and irrelevant. They're given names from bacterial proteins that they're not related to. And so I call that DMPK-CC. DMPK is this family of kinases. CC stands for coiled coil. So if we look at the diagrams, the domain diagrams of this set of, these are 6 of the 8, I guess, kinases, that's this, I don't actually like this color, I think I may fix this. The DMPK-CC is all in the middle, in the same color, given the same name, the residue numbers are given, the kinase domains are here in orange, and some other domains are out here at the C-terminus. So the colors are the same across all of the structures of this small family of kinases. If we look at some of the other sources, here's our domain diagram up at the top for CDC42BBP, BPB. And here's that CC domain in the middle. ECOD has this CHMP3 related. This is some bacterial thing that is just not correct. TED is another source from a database called CATH, C-A-T-H, CATH. It doesn't have anything for the CC region. It also doesn't have anything for this beta propeller up here. It's completely missing that. And if you go to the Uniprot website, it doesn't have anything for the CC region, and the other regions are the same. So these 3 sources are sources you could use, and ours is kind of structurally correct because I stared at the AlphaFold model and named everything with consistent names. That ECOD database is missing about 3% of our assignments. TED, which is what you get on the AlphaFold database, is missing almost 7%. Uniprot is missing 17%. So Uniprot is a bad source for domain interactions, and TED is more problematic than I would expect. Okay. We found 17 different domains in 50 or 60 kinases that are novel, that don't have names, that have never been given a name, and that have folds that are not related to anything in the PDB that we know of. There is a website called FoldSeek where you can give it some coordinates and search all 200 million AlphaFold models that are in the AlphaFold database and see, is there anything similar to that structure? I know you can't read this, but if I give it this magenta domain from a kinase called ALK, the first two that score 1.0 here are from different versions of ALK. All of the other ones are 0.12, 0.10, 0.06. They're from all sorts of other genes, but they're not at all similar to this structure. And so only ALK has this domain, as far as I can tell, of all of the genes in all of the sequences that have ever been deposited in UniProt. So we call that a novel domain, and I'm just calling it, ALK and terminal domain, because we don't know what the function is. We can use our ipSAE score in an intramolecular fashion of domain to domain. So here's a protein called NEC1. Here is the kinase domain. There is a small alpha helical domain. This is one of the novel domains down here. And the score between that domain and this domain is 0.45 on our score. And that's pretty good. That means that it's very likely that this small alpha helical domain interacts with the kinase domain, perhaps inhibiting it. Let's skip that. We can use our domain assignments. And this is for most proteins, you can go online and find a dozen or 100 different proteins that might interact with your protein of interest. We can make models of that full-length kinase, 2,000 amino acids, and all these other proteins that might be 500, 1,000, 2,000 residues long. Our ipSAE score is very good at figuring out, oh, that's a true interaction of these two domains and everything else is irrelevant. It's better than ipTM. But we also have the domain assignments for all the kinases, so we can say, oh, this is a kinase called Cask. It's a pseudokinase. So that's the kinase domain here in orange. This other protein interacts with the kinase domain. So this second protein is called DLG1. It interacts with something called the L27 domains here in sort of light blue. It interacts with one of them. This next protein, Lin7, interacts with the second L27 domain, and this last one interacts with the PDZ domain. So now that we've got these other proteins that interact with our kinase, because we know what the kinase domains are, what they're called, what maybe something about their function, we can say, oh, this protein interacts with this particular domain in our kinase. Perhaps the function is related to inhibiting that domain or changing its function or changing its location, something like that. So we're merging our protein-protein interaction information with our domain assignments together as one project, and we will do this for all 480 proteins that have kinase domains. I want to finish up quickly here. I talked about protein design, where you can have a target protein, and with RFDiffusion is a program that does design of small mini proteins, little helical bundles, to a target, and you can use our ipSAE score to filter out the bad models from the bad sequences from the good ones. This is from my colleague, my friend Richard Bayless at the University of Leeds. So this is this kinase is Aurora kinase. He did the structures of Aurora that I've shown. These are a whole bunch of different designs, these little helical bundles that he chose to bind to a certain location on the kinase domain. So here's a whole bunch of them. Some of these are crystal structures, but all of them all include predicted structures. So he did a whole, 20 or so different sequences and designed them on the computer. He used our ipSAE score, and so here's the ipSAE score, 0.75, 0.64, 0.71. These all had good KDs. These are really tight binders, 1 nanomolar, 3 nanomolar, 22 nanomolar. When the ipSAE score drops to 0.42, 0.53, 0.56, now the binding is a sort of 100 or 200 nanomolar. There is one outlier. This one scores really well, but also has terrible binding. So 6 out of 7, the ipSAE score was pretty indicative of tight binding when it's good score, poor binding when it's a low score. So I just want to summarize. AlphaFold and programs like it can give you structures with near experimental accuracy, as well as doing protein-protein interactions, post-translational modifications, DNA-RNA complexes, small molecule complexes, and I haven't shown you, but different conformational changes. You can often, if you run AlphaFold 50 or 100 times, 50 or 100 models, you will see different sets of models that might be different from each other in some way. We've done that many times where, yeah, it turns out that's biological, that this protein has 2 different conformational states. or it has multiple domains, and maybe in one set of structures, the 2 domains are like that. In some other set of structures, the 2 domains are like this. It'll turn out, oh, this is the active protein, this is the inactive protein, compared to experimental structures where we know the answer. As I said, the programs struggle with small molecules when they are very different from anything in the PDB, either the small molecule or the binding site in the protein. That will get better. DeepMind claims it has a program that is much better, but it is not giving us any access to it because they are selling it for billions of dollars. We always do high sampling. We always do 50 or 100 models, not just 5. Some events, some structures are 10 or 20% of the structures will have that correct model, that correct interaction. But if you only get 5 models, you run it once, you get 5 models, you might not get that one. You might not get that high scoring model. And so you want to run it on your own computers if you can. I'm willing to help anybody who is listening online or here. If you want us to help you model a particular protein, I'm certainly willing to do that. And we can run calculations on our computers. The programs can sample different conformations, either within a protein or even between proteins, and again, run it 50 to 100 times. You can do interactome studies where you take your one protein and maybe there's 100 proteins that may or may not interact with it, but maybe you think that they might. You can score them, and our score is very good at determining the true ones from the false ones, or at least the ones that AlphaFold can detect, where you get a good high confidence model between the list of proteins and your target protein. Remember that AlphaFold-like models, like any structure prediction, these are hypotheses. They require some experimental validation as you're going about continuing your project, investigating some particular protein, where you might want to do a couple mutations, make sure that if you look at the predicted structure, you make a couple mutations in the interface to make sure that that changes the phenotype in a way that, in a way that you would expect. And then finally, protein computational design. Our score is turning out to be very useful for this. You can design small proteins that bind to your target protein, even a particular site on your target protein, maybe protein-protein interaction that you want to inhibit. These technologies have improved really tremendously in the last couple of years. So I want to acknowledge where all of my funding comes from, all from NIH. I'm in Fox Chase Cancer Center itself. These are the people that did the work. Joan Zizio did all the active kinase work. Pragya Priyadarshini has been doing all the domain interaction stuff. Bulet Fayezov did some of the early work on the AlphaFold models. Qifeng Shi did those autophosphorylation structures that I showed that are part of our set. And Vivek Modi did the early part of our kinase classification from a PNAS paper in 2019. And then these are my colleagues that I work with. So I want to thank you for your attention.